So, hello everyone. We are once again in the what we are reading blog or video blog of the South American Mycorrhizal Research Network. At the time that we publish this interview, this will be our interview number 63, 64, I don't know. Um, and I don't know if you have noticed it, but or biases towards mycorrhizal ecology. So for a change, I wanted to do this interview with Professor Sebastian Schornack from Cambridge University in the UK in the Science Body Laboratory. He and his team published this very nice paper that you can see in the screen in Cell Reports entitled Mutualist-pathogen co-colonization modulates phosphoinositide signatures at host intracellular interfaces. So hello Sebastian, thank you for being here. It's really an honor to have you and well, of course, your laboratory in the University of Cambridge is very well known. So thank you for being here. And please tell us, in the first place, what is the context of this paper? Is a project? The work of a student or a postdoc?
Yeah, thank you very much for having me. It's great to be able to talk about our research. So we're studying plant microbe interactions, but we don't only study mycorrhizal fungi, we also study pathogens. And in nature, you have both of these types of organisms obviously co-occurring together. And the overarching interest really in this study was like what happens when you have a plant colonized by beneficial mycorrhizal fungus, but also attacked by a pathogen at the same time. And this work has been led by my former student, PhD student Alex Guyon, who's now a postdoc at the Boyce Thompson Institute with Maria Harrison's lab. And he basically started working with two organisms. One is Funeliformis mossae, which is a common arbuscular mycorrhizal fungus that forms arbuscules inside cells very nicely. But he also worked with Phytophthora palmivora, which is an oomycete, also filamentous, but not related to fungi. And oomycetes also form intracellular structures. They are much smaller. They have these little haustoria here that stick into the plant cells. So the question that we asked was very simple, like what happens if you have a beneficial fungus in the cell and then subsequently that cell is invaded by a pathogen? And because both of the structures that both of these organisms form inside cells are surrounded by a membrane, uh, the question was, are these membranes the same? Are they different? Are they changing? And so we took benefit of a nice system of reporters that was established before already in Arabidopsis. And this reporter system labels phosphoinositides. Phosphoinositides are components of membranes, and there are different types of phosphoinositides, PI3P, PI4P, PI45P2. We specifically focused on PI4P and PI45P2 because they both localize or are present in the plasma membrane. And so we use proteins that we express in the plants that have a lipid binding domain that's specific to the specific phospholipid, and then a fluorescent protein fused to it. And so we can see exactly where is the phospholipid in a plant cell. So in Arabidopsis, this has been tested with pathogens in the past. You can't really test it with mycorrhiza because they don't have that. So we actually switched to Nicotiana bentamiana, which is it's our lab model plant system where we have established both mycorrhiza and pathogen infection. And and so in this next image, you can see that well, I hope you can see, yes, that with this PI45P2 marker, which labels the tips of mycorrhizal fungi, we can make amazing animations. We're able to capture live in 3D how arbuscules inside living plant cells actually form. So each of these frames of these videos is like 15 minutes apart. And so this branching of these arbuscules is quite rapid. And because this marker labels specifically the tips, we know exactly which parts of the hyphae are growing into arbuscules, and we can put our microscope on top of that and just do time-lapse imaging. This is quite remarkable um, to see this development of structures live.
Can I ask you
Sure.
if this has been done before, in particular this kind of uh,
Live imaging.
live imaging of arbuscules?
So uh, Jen McGaley has
Mhm.
established a system called AM Slide,
Yes.
which allows the live imaging of arbuscules. By the time when we did the study, she has imaged in a time frame of days rather than minutes. So she hasn't really demonstrated this fast-paced dynamic, but by now she's doing that as well. Basically, her setup, this AM Slide setup, allows all of this imaging. Of course, it's important to actually you need to know where to look, right? So I think the advantage of this PI45P2 marker was that we see early on where the arbuscules are growing. Um, so to capture these early time points, you need to have such a marker. For later time points, when you already have arbuscules and you want to study their dynamics, then you can just do microscopy on a developed arbuscule, and you can see how long it lasts and when it disappears, etc. So all of this is possible thanks to this work from Jen McGaley's, from Uta Paszkowski's lab, which I cited here at the bottom of this slide, actually.
Thank you.
So
Yes.
definitely check it.
Yes, I remember her work from from the last icon. I think she got an award.
Yes,
Yeah.
yes. So we use these phospholipid markers to check first avascular mycorrhizal structures, and this PA45P2 marker here labels the tips of invasive hyphae and also the tips of arbuscules. It looks like that, and basically only the tips are labeled. The other phospholipid PI4P labels everything like a glove, surrounds both the arbuscules equally as well as hyphae that go through plant cells. So these two phospholipids are differentially distributed across avascular mycorrhizal structures. However, when you look at the pathogen, in this case Phytophthora palmivera, Haustoria, we see a different picture. Instead of just labeling the tips, PI45P2 labeled all of the Haustorium all around, versus PI4P, which is present at avascular mycorrhizal structures is completely absent actually from Haustoria. So it's really like completely gone here from these Haustoria. And and this was a phenomenon that was seen before with other pathogens that have been studied in leaves, for example, in Arabidopsis, that PI4P is excluded from Haustoria of fungal pathogens, and now in our case also of oomycete pathogens. So you can see here again, this is a root cell that has been invaded by Haustorium, and there's really no PI4P label around the structure, despite the fact that it's surrounded by a membrane. However, when you have an haustorium that's projected inside a cell that has an arbuscule, then you can see out of a sudden this label appears, this PI4P label appears. So the identity of this membrane has changed. It now has PI4P. And and it's not only PI4P, it's also other proteins that change their localization. For example, you can see here a myristoylated protein. This is a fluorescent protein that has an N-myristylation motif to be able to anchor to membranes. And normally these proteins are excluded from Haustoria. They are present at the plasma membrane. They're excluded from the Haustoria. But in a cell that is mycorrhized and tissue that is mycorrhized, you can see out of a sudden that these proteins with myristylation anchor not only label the plasma membrane, they are also co-occurring at the membrane around Haustoria. So every time there was mycorrhized tissue and Haustoria are projected into that tissue, the membrane around the Haustorium has changed. And that membrane is crucial for the pathogen because it needs to get proteins across this membrane into the host cell to suppress immunity, to manipulate the host. And so we were wondering if the membrane is changed, does this actually affect the ability of the pathogen to infect? And and so we did an experiment that many people have done in the past with other pathogens, and the result is similar to observations before. So we took two plants. One is non-colonized
Mhm.
and one is colonized. Here you can see that by the activation of the mycorrhiza-specific phosphate transporter. So these plants have colonization. And then when we quantify subsequent infection with a pathogen, here the elongation factor of Phytophthora palmivera, we can see that in the non-AM colonized plants, we have a lot of biomass of the pathogen. So this is very susceptible, and the pathogen can infect very well the root system. In the plant that has been pre-colonized with avascular mycorrhizal fungus, we see that the biomass of the pathogen is much, much smaller. So indeed, pre-colonization of mycorrhizal of roots with mycorrhizal fungi renders those root systems more resistant.
Mhm.
There are multiple reasons for that, but one of these could be because the cells that actually invaded by the pathogen are different to when they have been pre-colonized by avascular mycorrhizal fungi. So, so in summary, basically, um, you have these individual decorations of the membranes. This one here, PI4P, is present around the membrane. Here it's absent. But when you have them both in the same cell, then this decoration, this PA4P, also occurs at this Astoria. And I think we're the first ones to show that these phospholipid signatures can dynamically change depending on what's going on in the cell. And so you cannot just do a single snapshot of single interactions, one microorganism and one plant. You really have to also look into more complex scenarios and which are the ones that are actually out there, right? No plant is colonized by just one fungus or just one pathogen.
Sure.
So that's the take-home message, basically.
Well, that was excellent and very outstanding work. Let me ask you, so how fast can be like in real world conditions this change in the wall in the cell wall of this protein specifically?
Yeah, so haustoria are formed very quickly. So you have a pre-colonized tissue, and then the pathogen puts in haustoria, and right away when the haustorium is put in, the membrane already looks different. And haustoria are formed very quickly. They take like 20 minutes.
Oh
They're
wow.
not as big as our vascules, so they're sticking their fingers very quickly inside the host cell. And right away they have this different decoration.
Wow.
So there's no reorganization, or at least we cannot capture it with the imaging that we have. But you can take this further. You can use genetic manipulation now of cells. You can express enzymes that change phospholipid signatures, and then you can see what impact that has on the interaction.
Sure. Sure. So I think that uh, sorry, the other question will be if do we know the genes involved in these proteins, in these two proteins specifically?
Um, so the phospholipid signatures are controlled, for example, by enzymes that are phosphatases. Um, or kinases. So kinases add phosphates and phosphatases remove phosphates from these phospholipids. And this has implications on the membrane decoration. And when you look in genes that are differentially expressed during colonization with mycorrhiza, you find these type of enzymes differentially expressed. There's many of them, several of them. It's not just a single one. So you would have to go one by one
Okay.
using genetics to now address which ones are the ones that are relevant.
Maybe there is also epistatic interactions between these genes.
Absolutely, yeah.
Um, well, this is really amazing work. Because it's very similar, I guess, with recent ecological work, like by Marcel van der Heijden. They did this study, which we have the interview in the same channel. Stephanie Lutz is the first author, and they inoculated Rhizoglomus irregularis around 54. corn farms in northern Switzerland, and they found that the best predictor of a successful inoculation, which was found just in 25% of the farms, relatively low number, was those farms that had a really high abundance of initial abundance of plant pathogens.
All right.
Yeah.
Yes. Yeah.
So I am seeing that the field is going more into that direction, and as you said, of course, in the real world, this is what would be the most common situation, having all these different uh, fungal guilds interacting at the same time. Well, thank you so much. This was really sorry, and one last thing is that I'm really
Yeah.
amazed on how fast this is. So it tells me that there is a lot of phenotypic plasticity going on, that this symbiosis um change really quickly according to the interactions that are going
Yes.
on.
Yeah,
Uh,
we have seen an empty cell that on the next day had an arbuscule and on the next day was empty again.
wow.
So, so normally arbuscules live 2 to 5 days, or that's
Yeah.
what literature says, but
Yes.
you can see all sorts of variation in that. So sometimes it just takes less than a day to form a full arbuscule and a day to get rid of it again, right?
Oh
And
wow.
that's a level of dynamic
Yes.
that we don't really capture when we take out roots and stain them, where we kill the roots and stain them. I think this type of life capturing the dynamic system is quite, I would say, an imaging frontier, but it's being developed now, so new possibilities open up.
Well, and that has a lot of implications for many debates going on in our field. For example, the people debating the percentage of plants that are arbuscular in a global scale. Some say that you can only check arbuscules. Some say that you can check other structures. But if they form and die so quickly, it's challenging. I myself have studies in here in Chile where we found really negative relationship between phosphorus and percentage of arbuscules
Mhm.
um, in a conifer or conifers here are arbuscular mycorrhizal. Uh, but now you make me question. Oh well, if this is so quick, if this is
Yeah,
so
well,
quick.
we actually used on purpose Funeliformis mossae because in Nicotiana bentamiana in our hands, this forms a massive amount of arbuscules and very little vesicles.
Ah,
So we actually get the structure that we desire the most to image
perfect.
most abundantly, right? Whereas with Rhizoglomus, as you said, we see much more vesicles and much less arbuscules, especially at late stages.
Yeah, of course, there should be some phylogenetic signal across the AMF
Yeah.
genera. The last question is, what are you working right now, either in this topic in specific or
Uh-huh.
other topics?
I think in the field of symbiosis, my research, my frontier research is really on effectors, on mycorrhizal effectors. A while ago, we have identified that there are some proteins encoded in the fungus that are also known from pathogens, and we think that they help the fungus to colonize the plant. We still have to figure out how exactly that works, but we think they're there for a purpose, and they're actually secreted by the fungus in the host tissue to manipulate the interaction. This sounds a bit like a pathogen interaction, but I think also in symbiosis, and this has been shown in the past by others, effector proteins play an important role, and they are much understudied in mycorrhizal symbiosis, and will trying to contribute to that.
Oh wow, that sounds really exciting. Well, thank you so much for being here, and I hope to see you maybe at ICOM or other conference.
Yes, I'm not coming this year, sadly, to ICOM. It's a bit far for me, but I hope to see you at another conference or at the next ICOM.
Yeah, of course. Thank you.
Yes.
Bye bye.
Bye bye.